Hi guys, welcome back to Recombinant DNA technology, and today we're going to cover about methods in gene cloning. One of the successful story about gene cloning is producing this adorable baby monkey which can glow in the dark. What about having this ability in yourself? Maybe now you won't need torchlight in the dark, or maybe you can scare the ghost away. That would be good, right? So for today's learning outcome, you're supposed to be able to know about the methods in gene cloning. So we're gonna look at the overview using the diagram to show the step in gene cloning using plasmid. Before that, let's check out this terminology. Gene cloning, it is the production of exact copies or clones of a particular gene or genes using genetic engineering technique. Just like this Naruto here with Kage Bunshin no Jutsu, he produces exact copy of himself, but in this terminology, we're gonna copy specific genes. So there are 5 steps in gene cloning by using plasmid as vector. Step 1 is isolation of genomic DNA. Step 2, cleave. Step 3, insertion. Step 4, transformation and amplification. Step 5, screening. So before we go deeper into it, let's check out the concept. So the first step is to isolate the genomic DNA, which is the bacterial plasmid, as the vector, and also the foreign DNA which you wanted to clone. So you take out this plasmid and then take out the gene of interest. The next step is to splice with restriction enzyme. So we use the restriction enzyme to cut the restriction site so that the vector can be opened, and then you can put in the DNA fragment of interest. The next step is the insertion. When you have already cut open the plasmid, now you may insert the DNA of interest. And the next step, call it as transformation and amplification, which actually make introduction of cloning vector or recombinant DNA into bacterial cell or host organism by cloning of target gene by the host cell. Due to plasmid having origin of replication, the plasmid itself with gene of interest may be cloned by itself inside the host. At the same time, the bacteria as the host may as well reproduce and make copies of themselves alongside with the plasmid inside them. So now you amplify it in like 1,000,000 times. So the last step is called screening. Why do we need screening? Because you will produce 2 types of plasmid, whether it will become recombinant DNA or non-recombinant DNA. So how are we going to screen it? We're going to use the genetic marker that have in the plasmid, which is the ampicillin resistant and lac sac. What will happen is it will form blue-white colony, just like this one. So which one we're going to take? It's gonna be the white one. Why? We're gonna look deeper into it soon. So let's go back to our learning outcome. Let's check our overview, the method in gene cloning by using plasmid as vector. Why plasmid? It is because it have good characteristic, which is it have the origin of replication, enabling it to replicate independently inside the host, and it have multiple cloning site, or you call it as polylinker, which enabling it to accept foreign DNA by cutting this site. You can use Hindi, you can use BAM-H1, you can use MA1 or EcoR1 to cut this plasmid and then to insert the DNA of interest. And what good about it as well is because it have the marker, genetic marker, which is ampicillin resistant and lac sac. And do note that the lac sac is where the polylinker is. So when you disrupt this and insert something in it, it will become mutated and won't be able to function as lac sac anymore. Instead, it will produce the gene of interest that you insert in it. So let's check out the step. Step 1 is isolate of genomic DNA, the gene of interest. So here we take it from probably human DNA, then take it out. And then also we need to isolate the plasmid as vector. So you can take it from E. coli because E. coli have accessory DNA called plasmid. And why plasmid is chosen? Again, because it have the ampicillin resistant and laxi as the marker, also origin of replication, and it have the restriction site here where you can cut it. And then the next step is called cleave, where we're going to cut both plasmid and gene of interest with the same restriction enzyme. Remember, at LAGZAC there is the restriction site for the restriction enzyme. So we're going to cut it here, and then we're going to put the gene of interest in here. The next step is called insertion. When you have cut it, you may now insert it. And you must add DNA ligase, where this DNA ligase will form phosphodiester bond, hence making the plasmid functional. Although in theory we hope that we have recombinant DNA plasmid where the plasmid take up the target gene. However, in reality you will produce two variants, whether there will be insertion or there will not be the insertion. So the one without insertion, you call it as non-recombinant DNA, and while the one with insertion, you call it as recombinant DNA. This is the product after insertion. So we're gonna take this one only, and that's why we're gonna do the screening later on. The next step is transformation and amplification, whereby we're going to put all the product here into a host. So the host is something that will accept this DNA, because if you doesn't put it in the host, this DNA will do nothing. Instead, in the cell or in the host cell, then it will produce the protein that we wanted or the product that we wanted. So we must put it back in the host. So how are we going to do that? It is called as transformation. Basically means recombinant DNA plasmid. We're gonna put it into the host, and the example here is E. coli because we took the plasmid, the vector, from the E. coli, so the perfect host would be the E. coli itself. Hence, you will get two types of hosts, a host with recombinant DNA and a host with non-recombinant DNA. So that's why we need to undergo the next step, which is screening. So in screening, we're going to plate the cell onto medium with ampicillin and excal. Why we're going to use ampicillin? It is because this plasmid that we choose have ampicillin resistant gene. The host that does not take up any of the recombinant or non-recombinant DNA, the host will automatically won't survive on this plate. The next one, identify it by using the LAGZAC gene that have been disrupted or the LAGZAC gene that will not be disrupted. If it is a recombinant DNA, the gene of interest will be inserted in between LAGZAC gene. Hence, LAGZAC will not be function anymore, so it will not break down lactose. It will then appear white. So these are the white colony, means it contains the recombinant DNA. So the host cell here contains recombinant DNA. While the host cell that have non-recombinant DNA, which means it does not take up the gene of interest, it's sealed back, hence it will get its ability back, which is LAGZAC gene. It can break down the lactose, so it will appear blue, just like this one. So which one will we further culture? Would it be blue or would it be the white one? So the one that we will further culture will be the white one, because it means we have the recombinant DNA there. In screening, only a cell that took up a plasmid which has ampicillin resistant gene will reproduce and form a colony. Colonies with non-recombinant plasmid will be blue, because they can hydrolyze excal since the LAGZAC gene is not disrupted. Colonies with recombinant plasmid in which LAGZAC is disrupted will be white because they cannot hydrolyze excal. So this colony, the white colony, will be selected. So again, step 1 is isolation of genomic DNA of interest and also taking out the plasmid, because we need the vector, we need the one to carry the DNA of interest into our host. Then the second step is to cleave both gene of interest and vector with the same restriction enzyme. To the step 3, which is insertion, whereby we insert the gene of interest into the vector, which is the plasmid. Step 4 is transformation and amplification, whereby transformation means we introduce the recombinant DNA into host cell, and then it will clone itself inside the host cell, and also the host cell itself will clone themselves alongside with the DNA. And lastly is screening, which colony that actually have the recombinant DNA. So that's all for now. See you next lecture.